plant phenomics community here at Iowa State is using every technology that is at our disposal, including robots, lasers, drones, 3D imaging, and machine learning. You could almost say that the only technology from Star Wars that we aren't using is the Force. Although I'm pretty sure I've heard Dr. Yu refer to quantitative genetics as being the Force. My first semester here, I walked into Danny's office with my project proposal in hand. I was going to fall into Race and Kyle's footsteps and work with drones, thermal imaging, and hypospectral cameras. And Danny, he sat me down, all fatherly, and said to me, "Kevin, I think you should work on roots." Well, I didn't know this at the time, but it was an opportunity in disguise. And I'm glad he gave it to me. The root community is close, a tight-knit, and really growing. And it's really allowed me to turn colleagues into friends. Just go on Twitter and hashtag root or hashtag root biology, and you'll see numerous comments and threads pop up. I came to Iowa to learn, but not only did I learn, working meticulously on these stubborn temperamental roots really helped me build character. So in my project, we selected a diversity panel of 292 soybean genotypes from the USDA core collection, and these have already been genotyped, with aims to identify the genetic drivers for root system architecture traits. In the interest of time, I won't go into great detail on the methodologies we created. This growth chamber system that we developed was able to grow and image up to 400 seedlings simultaneously per day. And this allowed for repeat imaging. One of the main shortcomings of root research is applicability. I wanted to create a system in which we could correlate our lab results with field-grown roots, and then use genomic prediction to identify genotypes of interest from the USDA core collection of 19,000 accessions. This way we could better implement our workflow into a breeding pipeline. We built a mirrored approach in the field as what we had in the lab. We grew our panel of plants in hill plots and removed them by hand with shovels. And this was a lot of work and gave us only 4 plants per genotype from 2 sites, Ames and Muscatine. Learning from this, we were able to move to more of a mechanized approach of root removal. After removal, the roots were washed and imaged using one of 2 imaging stations. We did this both 3D imaging and 2D imaging, as you can see here. Before analyzing the root data, I needed to look at the genomic information to understand the relationship of this diverse panel that we created. So I built a dendrogram using the SNP data that helped visualize these relationships. To aid in the analysis, I clustered the genotypes into 8 groups based on their SNP data, and these are the branches that you see here on the diagram. I found it interesting how genotypes from certain countries are related, and you can see this in the leaves. You can see the American-derived genotypes in magenta in the top left-hand portion of your screen. That's all genotypic cluster 1, and all but one of the American genotypes actually falls within that genotypic cluster. You can see the same trend with many of the other countries that we are using genotypes from, including Japan, Russia, and Korea. And China just has the background itself in which we built this system. Next was to start to affiliate the genotype with the phenotype. Were there certain clusters that were good? Maybe regions within the clusters? How were we going to look at this? We couldn't look at it really genotype by genotype. So I clustered the phenotypes based on their trait values, very similar to how I clustered the genotypes based on hierarchical clustering. So here the leaves of the tree, the text, display the phenotypic clusters for each genotype. For example, gray is phenotypic cluster 1 and yellow is phenotypic cluster 8. Now, roots that had the higher root traits were automatically clustered into one of the early phenotypic clusters, 1, 2, or 3. The poor performing roots were subsequently clustered in the 6, 7, and 8 groupings. Clusters 2, 3, and 5 were generally pretty good, while clusters 6 and 7 were generally pretty bad. Most of the clusters had some good and some bad genotypes. However, in discussions with some of my lab mates, I remember John had said to me, well, what is good? And I responded, well, high total root length is good, or a large number of roots is good, or a long taproot is good. But maybe it isn't. Danny had mentioned that maybe I'm not thinking like a plant breeder. After all, we mold plants to fit their environment wherever we're breeding for. We need these breeding objectives. And that got me thinking, well, I'm really curious to where these genotypes were originally adapted. What type of soils are they from? What type of environment? What type of climate? So after doing this analysis, I took a step back and decided to change course to something a little bit more, I don't know, biologically relevant. I created 5 root types based on previous work that I found in literature, highlighting features that was maybe 4 different environments. For example, the foraging type was produced to efficiently scour the subsoil to extract immobile nutrients in poor fertilized soils or low fertility soils, while the drought-tolerant type was built on the steep, deep, and cheap idiom, in which a steep angle of root attack into the soil would then prove to go deeper within the soil and chase the water and the nutrients through the season. So in order to align the genotypes that we had collected and these different root types, I built an index for each root type comprising of multiple traits that would build these types, and gave each genotype a rank for each root. For example, within my index for drought tolerance was angle, and a steeper angle got a higher score. The roots that I show here on the paper, or on my slide, are the genotypes that scored the highest for each of those particular root traits. So you can see that the drought-tolerant type has very steep angle, whereas a foraging type is quite prolific and far-reaching. The umbrella type looks somewhat like an umbrella, and the beard type, well, doesn't look all that great. The maximum type is essentially just built out of 7 different traits, and the maximum of those 7 different traits, including growth rate, the number of roots, the total length of the roots, the convex area of the roots, and so on. Now with these 2 approaches, both the phenotypic-based clusters and the root types, I looked again at the data. In this heat map, the genotypic tree is on the far left. Now it's not a circle anymore, it's in a linear fashion, but the colors remain consistent, with the American cluster being in magenta again. Now the root types are ranked with the black and the red next to that. Black being highly ranked or positive, and the red poorly ranked. Moving on, still I have 13 root traits that make up the root type indices and their performance, the blue being high performance and the orange being low performance. So trends started to form, but I find it's easier to look at this graph sideways, so hold on, let me modify the code. Alright, I like to think about this as matrix style, so look straight down the page. Let's first look at clusters 6 and 7 on the left-hand side. They are red and orange, the red being lowly ranked root type and the orange being poor performing phenotypic type. Next, there is a subbranch within cluster 2, the left branch, which is very strong, including a black streak, a dark black blue streak, and then below that a blue streak within the phenotypic traits. Other traits within genotypic group 2, the orange group, perform well, but not nearly as well as this one branch off the left of this main tree. In other regions perform well too, especially in phenotypic or genotypic group or cluster 3, the yellow cluster, which performs well for both the root type as well for the phenotypic values. Other regions that perform well are the right half of genotypic base cluster 5 as well as genotypic base cluster 8. Cluster 4 produces a lot of beard type roots, but as you've seen that the beard types aren't especially strong growing plants. Now cluster 1 is made up of American genotypes and isn't particularly good or particularly bad. However, this cluster did have the longest taproot, and this is a result that I'd like to follow up in the field in future studies. Anyway, I could talk about these results for hours, but I'll move on. Our field analysis is ongoing. However, I did take some time to identify the top ranked roots for some of the root types. Just a note, these roots on the right, the R8, are hanging from the growth imaging chamber and droop due to gravity. So don't think that that's actually the way they look within the soil. As you can see, in the V1 and R8, the maximum root type is very prolific, having large and many long secondary and tertiary roots. The foraging root underneath it stretches through the soil with long secondary tentacle-style roots, without having substantial tertiary roots. Unfortunately, the V1 stage, we're unable to capture the root angle because the root is so young and doesn't have a rigid structure. However, I am interested if the root angle in the drought-tolerant types correlates between the lab and the field. That would be very interesting. I hope that our future studies will find evidence of lab to field correlation. Lastly, we were able to do some 3D imaging. Here are some of the image results from that 3D imaging of those same roots that we were looking at. We used a system called projected light, in which we shone projected light from a projector lines on a root and the camera picked up how those lines of light bend around the root, similar to if you stuck your hand in front of a projector and you watch the light from the projector bend around your hand. It's promising. Our results are somewhat inconclusive. We're still looking how to take these point clouds of data and really extract root length and root angle from them. But we're excited moving forward in this project. Anyways, thank you for your time, and I look forward to discussing this with you further. Come to my poster. I'm presenting it shortly after at this first break, and you can learn all about this stuff. I'd be happy to explain it to you.